actually in the lab, in the actual experiment, okay, I always tell my students that I don't expect them to get the um, accurate result, especially if like it's the first time to do experiments and all that. Contamination is number one culprit there, that you will not be able to get the ideal result. And also, aside from contamination, is the preparation of those enzymes. You never know, the lab techs may not be preparing those enzymes properly, or they should be stored in a cool place and they were not, the lab technician forgot to put them in the fridge, something like that. Okay, so some technicalities also happen in the actual. So we are trying to simulate, okay, or like as I've told you, just to comply with the requirement that you will be able to do the lab report. You just have to listen, and that's like what we can discuss properly. Okay, don't sleep. Anyway, okay. So you also have uh, supporting materials, the fish eggs, if you have that, the lobster. Okay, so we can do that if we have more time, but I will be more focused on the discussion per se for your lab report. Okay, so please bear with me. So this is chapter 29, a virtual lab, basically. Okay, so this is a table. All of these PowerPoints, you have them in the table, as you know by now. Excuse me. What happens? Okay, this is a flow chart of digestion and absorption of foodstuffs. Okay, so we should have a at least a bird's eye view of what happens to the. So like if you you purchase a Burger King, if you love burgers, okay, where is the carbohydrate there? The bun, right? What about the protein, the meat? Okay. What about the fat there? That's the of course the fat is sandwiched with the meat. Okay. You know, burgers are very fatty. Also, they have a lot of oil. So it's like basically what happens to the food that we eat. Okay, now if you're a vegetarian, you also have carbohydrates, and plants are have carbohydrates, and plants have like the starch. So this is carbohydrate that we get from plants or vegetables. Okay, so so this one, carbohydrate digestion. So the foodstuffs are stuff and disaccharides, they will be converted. This is what I was telling you, what will happen to the food that we eat. So it will be digested all the way down to the glucose level, okay? Galactose, glucose, fructose, these are the simple sugars, okay? Simple sugars that will be produced when we digest starch and carbohydrates. So what is the role of the enzymes now? So we have salivary amylase, okay? The salivary amylase, it comes from the salivary glands. What are the three salivary glands? We have the parotid gland, the submandibular gland, and the sublingual glands. Okay, they produce salivary amylase. Aside from enzymes, of course, they produce saliva. Another enzyme is pancreatic amylase. Of course, this will be coming from the pancreas, as the name implies, pancreatic amylase. Okay, so that's an enzyme produced by the pancreas. It's to help in the digestion of fat. So as you see, the last name there is amylase. Amylase, salivary amylase, pancreatic amylase. Where you find them, of course, uh, this one, salivary amylase in the mouth. You see my pointer? Okay, this one. See my pointer? Salivary amylase. Okay, thank you, Rinchan. In the mouth, okay, and of course the pancreas is the uh, that secretes pancreatic amylase, and where is the site of action in the small intestines. As you saw when we did the anatomy, if you remember the pancreatic duct and the cystic duct, it will open in the duodenum. So it's like the action of this amylase, of course, will be in the small intestine. Definitely, if not, it will not go up to the mouth. Okay, so pancreatic amylase will work in the small intestines. Well, the path of absorption, for glucose, galactose, they are absorbed via the co-transport with sodium ions. Okay, fructose passes via the facilitated diffusion. Okay, and all the monosaccharides leave the epithelial cells by facilitated diffusion. They enter the blood. Okay, so here this is absorption already. Okay, absorption. So mostly some are absorbed or digested in the mouth right away. That's why you feel or you taste sweet already when you try to chew carbohydrates or bread, right? You taste the sweet taste because it's like it's been digested already right there in the oral cavity. Okay, but mostly, of course, digestion is in the stomach. Now, for the absorption, of course, for the small intestines, when these monosaccharides here, um, when we were able to digest them, then they will be absorbed. Okay, they will be absorbed by the mucosa of the small intestines and then be transported to the blood, to the capillary blood, and transported to the liver through the hepatic portal vein. Okay, well, if you remember, we discussed like the hepatic portal vein, everything that is absorbed. That's why it's the liver that is like this, the screener or the quality control, or what we call it detoxifies whatever is toxic in the food that we take in. This is through the hepatic portal vein. Okay, so that is what happens in the absorption in the small intestines. See the brush border enzymes in the small intestines: dextrinase, glucoamylase, lactase, maltase, sucrase. All of this, okay, will be in the brush border of the small intestine, so that digestion of these carbohydrates will be faster, okay, to convert them into sucrase, so it will convert it to sucrose. Okay, maltose, the ASE, these are the enzymes. Okay, enzymes that will dissolve whatever is in the is in the brush border of the small intestines. Okay, so all of these amylase, lactase, maltase, sucrase, they are the enzymes that will help in the absorption of carbohydrates. Okay, next. Next, protein. Okay. The next we will digest is protein. It's like you imagine that's the meat in the burger. Okay. So what is the enzyme? Okay. What is the enzyme? Pepsin. Okay. Pepsin from the stomach glands. Okay. Also we also discussed there are cells in the stomach that produce hydrochloric acid. Okay, then binding with hydrochloric acid, the pepsinogen, if you remember. Okay, if not, you review that pepsinogen, and then combined with hydrochloric acid will produce pepsin, and this pepsin is the one that will okay, that will dissolve the protein, okay, into large polypeptide. If you remember in your bio, okay, polypeptide. When you say polypeptide, these are still large proteins, okay, large proteins that are from the digestion of this meat that we take in. Okay, other enzymes are pancreatic enzymes. Trypsin is a pancreatic enzyme. So it's like basically you remember what are the enzymes for carbohydrates, what are the enzymes for proteins, and so on and so forth. So for proteins, you have pepsin, you have pancreatic enzymes. These are trypsin, chymotrypsin, carboxypeptidases. When you say pancreatic enzymes, of course, they are produced by the pancreas. So when produced by the pancreas, you know that they will be um, acting in the small intestines. Okay, they will be acting in the small intestines because you know the pathway of that pancreatic duct again, opening up in the duodenum. So it will not back up to the stomach. You know the duodenum is the first part of the small intestines. Okay, so now you have the proteins, still broken down by the enzymes. Okay, to small polypeptides, okay, and small peptides, and now in also in the brush border of the small intestines, aminopeptidases, carboxypeptidases, and dipeptidases. These are the enzymes in the small intestines now. So to break down all of those protein or peptides into amino acids. Okay, so if carbohydrates, the end product are the monosaccharides. For proteins, it is the end products. Protein digestion is or are the amino acids. Okay, some dipeptides, some tripeptides. So the peptidases from the intestines will also help in further digestion. Okay, so this is what happens in the protein that we take in. So mostly absorption. Of course, the last column there is always absorption. So in the stomach, what happens? Amino acids, they are absorbed via co-transport with sodium ions. Okay, small dipeptides, tripeptides are absorbed via co-transport with hydrogen and hydrolyzed to amino acids within the cell. So that is like basically what the pepsin do and hydrochloric acid. Okay, there's some sort of absorption after digestion, some sort of absorption, but mostly the absorption is in the small intestines. That's why I told you the lead organ for absorption is the small intestines. The lead organ for digestion is the stomach. But some absorption in the stomach already occur. Okay. Now in the small intestines, um, infrequently there are transcytosis of small peptides occurs. The amino acids will leave the epithelial cells. Where will they go? They will be diffused. Through facilitated diffusion, they enter the capillary, absorb again in the capillary, in the villi, and all transported to the hepatic portal vein. Okay, always like that in the digestion. So hepatic portal vein has a very big role in the absorption and in the circulation of the nutrients that we take in. Okay, so that's for the protein. Next is the nucleic acid. Wait, where's the fat? Well, I didn't see the fat. There's the protein. Okay, this one. This is the fat. I thought you're missing the slide. These are in your content area, and so you may know. Okay, so for the fat, what is the fat that we have there? Well, maybe the olive oil, they still put in the veggie or something. Okay, so in the emulsify, when you say emulsify, that is to break down. Okay, to break down fat, emulsify. So what are these enzymes that are responsible for the emulsification of fat? This is the leader of the band. Okay, for emulsifying fat, it is the bile that is the leader of the band. But there are also other enzymes that will emulsify the fat. So look at that. In the mouth, you have lingual lipase. It says they're minor importance, minor importance. in the just so you know that when the food is taken in, there's something is already happening there. Okay, just a little bit. So then when we say, I told you this, the leader of the band, it's the bile that is the strongest link. Okay, that's the strongest um, molecule or substance that will emulsify or break down the fat. Okay, so it comes from the liver. It's the liver that produces the bile, and it is stored. The gallbladder is another accessory organ of the digestive system. Of course, the main organ there is the liver because it's the one that produces the bile. That's why we can live without the gallbladder, but we cannot live without the liver. Okay, so the bile is stored in the gallbladder. The function there of the gallbladder is to, if we need, let's say, for example, you ate too much fat, okay? So the gallbladder will have, like, there is a signal, a hormone that will make that gallbladder contract so that it will release more bile if we need it. Okay, so it says there, emulsification by the detergent action of bile salts. Okay, is adducted in from the liver. When I say detergent action, if if you wash dishes, you know what we mean. Like I don't want to say a brand. If you put a liquid soap there in a so oily water, when you wash the dishes, put the liquid and it will clear out the surrounding wherever that detergent is. So that is basically emulsification. Okay, so some sort of bile is like a detergent to dissolve the fat. Okay, now the for the pancreas also, it has a a role. The lipases, okay, lipases. So those are the enzymes that will break down the fat. Okay, so what will happen to the fat that we eat? It will be digested, the mouth and the stomach, the small intestines, and then to convert it into monoglycerides and fatty acids. Okay, this is the fatty acids. These are the end product of fat digestion. Okay, end product of fat digestion. Same thing, like the activity, they enter the intestinal cells, then the chylomicrons will enter the lacteals of the villi and transported to the system. This is more of like, maybe you will do this in your lecture, like for the discussion of those chylomicrons entering the villi of the small intestines. Okay, some sort of chain fatty acids are also absorbed and move into the capillary. Okay, so now we know what happens to carbohydrates, proteins, and fats. What about the nucleic acids? Nucleic acids are like coming from, like, basically also the breakdown product or digestion of like amino acids. Okay, now so for the nucleic acids, we have the pancreas, okay, and the small intestines. For the pancreas, pancreatic ribonuclease and deoxyribonuclease, and that will act in the small intestines. Okay, these units will enter in the intestinal cells by active transport. Okay, moving to the membrane, and then the units are absorbed into the capillary blood, again to the hepatic portal vein. Okay, hepatic portal vein. So, what are the end products of these nucleic acids? If you break them down using the pancreatic ribonuclease or the oxyribonucleases, and also the nucleosidases and phosphatases in the small intestines, they will break them down into pentose sugar. Okay, containing bases and phosphate ions. So these are the um, uh, end product, okay, end product of nucleic acid digestion. All right, so that is the basic knowledge
Also,
that we have to. Yes,
uh, what the function for the nucleic acids? Like, what they're doing?
what they're doing. You mean what is the function? So these are the nitrogen containing bases, phosphate ions. Doesn't ring a bell. It's the DNA and the RNA, production of DNA and RNA, okay, for the proteins, for the nucleus, okay. All right, so that's the role of the nucleic acid for the, of course, for the generation and regeneration of the cells in the body. Okay. All right. So following we have the slide here. This is the first activity one. Okay. Activity one is all about the salivary amylase digestion of starch. Okay, so in this activity, of course, the material that is present that is usually available in the lab are starch. Okay, so as I was telling you, sometimes technical problems arise, like the preservation of those starch, preparation, and all that. But we will have, of course, we will assume or presume that the materials here are freshly made. Okay, so this is just like to identify how the salivary amylase digests starch. Okay, so uh, just following, we have the test tubes here: test tube 1A, 2A, 3A, 4A, 5A, and 6A. So we prepared like about six test tubes. We have six test tubes here, and then we have this colored uh, additive key. So in this chart, we follow this additive key. If it has purple, it should be amylase, green for starch, white for maltose, and blue, light blue for water. Okay. So what do we have here in 1A? Test tube 1A, we have the enzyme amylase. Okay. Remember, the enzyme for starch or carbohydrates is amylase, okay, either salivary or pancreatic amylase. Okay, so test tube one is blue and purple. So it is what? What are our additives? Additives. Tell me if you understand what I'm saying.
Amylase and water, they say that.
Okay, you have amylase and water. Thank you. I just want you to respond so I know I'm not wasting my saliva here. Okay? Okay, 2A.
Water and starch.
So we have
Water and starch.
blue and starch and water. It's right there. And then we have blue and white, maltose and water. Okay, so meaning this one has an enzyme. This one has no enzyme. This one has no enzyme also. What about this one? 4A. We have amylase, the enzyme. We have to boil that enzyme. Okay, now I'm bringing you back to Bio 1. Okay, and the enzyme activity, if you remember, I want you to recall. If not, it's okay. What can we do? Senile. Well, anyway. Okay, so we have to boil. Why? What will be the effect of temperature on the enzymes? Okay, you should have a report. It's been a long time. I haven't been teaching Bio 1, but I still remember. So we have to boil the enzyme because we know that the enzyme work at an optimal temperature, meaning the enzyme will work at what temperature? Especially when we're talking about the body. How could we be normal? How could these enzymes in our body work if we don't have the correct temperature, right? So enzymes work at the optimum at the temperature of 37 degrees Celsius or centigrade, okay? At a normal temperature. Okay, if we were talking about the enzymes in the body, of course, what is our normal temperature? 37 degrees Celsius, right? So now, You boil that enzyme. What do you think will happen to that enzyme? It says there boiled amylase and then you put starch. So will this enzyme work? What do you think? No, okay? So at least you know the what's gonna happen, okay? So this enzyme will not work because you boiled it. You boiled it for 4 or 5 minutes. It's like you denature the enzyme, okay? That's the term. You denature the enzyme. It's like basically you're killing the enzyme. It cannot work, okay? It cannot do its job, all right? So then this is 4A with a boiled enzyme. And then this is 5A, amylase and starch, okay? And then this is 6A with amylase and starch also. What is the difference between these two? The temperature that you will incubate, okay? So putting all these additives, okay, so you know you can interpret amylase and water. What's gonna do? It has nothing to work with. It's not, amylase is not for water. Amylase is for starch, right? So what do you think will happen with 1A? Okay, you should put it in your mind. Now this one, starch and water. Okay, there could be some, starch also dissolved in water, but there's no amylase here. Okay, what about this one, maltose and water? There's no enzyme, and maltose is already a simple sugar, so there's not much to happen. What about this one? Your enzyme is dead. Okay, what will it do with amylase and starch? Okay, and amylase and starch, and then amylase and starch. Now these two will have a difference on this. The incubation condition. All of them are at 37 degrees Celsius except for 6A, which is so when you say incubation, the incubation here is usually 1 hour. Okay, so we have to check we have to check after an hour. Okay, so before I usually ask the students, okay, just to know if you are with us. So the incubation period is 1 hour. Okay, so the temperature, okay, is the one that will be different right here in 6A. If amylase and 5A is at 37 degrees Celsius and 6A is at 0 degrees Celsius. So all of these test tubes will be incubated or will be soaked in 37 except 6A. So there's a separate box for, we have an ice bucket there and put that test tube with amylase and starch at 0 degrees Celsius. Okay. And at 37 degrees, we put them in the water bath. Okay. If you did Bio 1, you also did water bath, and that water bath will be set to 37 degrees Celsius. Okay. Now, after the incubation period, what you're gonna do? So this experiment is basically filling out this table. Okay, you should have all these tables in your report. Okay, you should have these tables in your report and interpret. Okay, so well, anyway, what I'm telling you is what you will put in this table. Now, IKI. What is IKI? You should know. This is not new. IKI, iodine, potassium iodide. This is a test for what? Check that out. It could be in your test. Yes, there's a color. There should be a color change. Will there be a color change or no color change? You just have to put positive or negative. Okay. Your answers here are positive or negative. Okay. So let me ask you now. IKI is a test for what? Is it a test for
Chemical
starch
reactions.
or is it a test for simple sugar? Mm. It's a test for carbohydrates, right? Which carbohydrate? Is it the big ones or the small ones? IKI is a test for starch or glycogen. Okay, starch is a storage form of glucose in the plants, and glycogen is a storage form of glucose in animals. Okay, Okay, IKI is a test for starch or carbohydrates, okay, or what do you call this? Polysaccharides, okay, IKI. Now, so let's say, for example, we soak this for at 37 degrees at 1 hour. After 1 hour, we test it for IKI. Now, what is IKI? What is the color? If it's positive for initially, it will be what? There will be no color, and it will, yes, it will change the color. If there is sugar, okay, not sugar, sugar, of course it's sugar, but if there is starch, then it will be blue black. Okay, the color change should be blue black if it is positive. So, what do you think with amylase and water soak at 37 degrees? Will there be starch there? Will it be positive for starch? Will it be blue black?
No.
No, yes, no. I like those answers. You answer them, okay? So you understand what's happening. Okay, no. No. Okay. So where will starch come from? So therefore, it should not be positive. Okay. So your answer there, do you follow? Yes. Okay. Do you follow? So what will be your answer? Negative. Okay. Negative. There should be no color change because there's no starch there. There's only amylase and water. Yes. Yes. Okay. So what about this one? Starch and water, soak at 37 degrees. What will be your IKI test? Is it blue black? Blue black, okay? Because there's starch, presence of starch. Yes, yes, yes. Thank you. What about here? Maltose and water. Maltose is a what? Simple sugar. So IKI cannot detect that anymore. So which will be also, there will be no color change. It's negative, meaning it's negative for IKI. Okay. So your answer here is negative. What about here? We said that we boiled the amylase. You killed the amylase. So what will be your um, uh, test, IKI test for amylase and starch? Mm-hmm. Catchy, catchy. Is it negative? What are your additives? You have amylase and starch, right? What is the color? Okay, okay. I like that. Positive. Why positive? Because there's starch there. It was not dissolved. Okay? Because amylase is dead. It was boiled. You get that? Okay. So this IKI test should have a color change. So the color there should be blue black. Okay? So it is positive. What about here? 37 degrees centigrade, amylase and starch. There is an enzyme. There is starch, very good temperature. Po, what is po? Okay, it should be positive. The temperature is good. The amylase is there. Starch. Okay. Did the amylase work? Why will you say it's positive? IKI test is a test for IKI test is a test for starch. It's not a test for simple sugar. Okay? Okay, okay. So we have amylase and starch, and then with good temperature, after 1 hour, did amylase work or not? Mm-hmm. Amylase work. So if amylase work, what does amylase do? It dissolves or breaks down starch, right? So if you test the test tube for IKI, will there be starch there? Will there still be starch? Mm. Okay, so there should be no more starch. If we did this like in a optimal environment, very careful, no contamination and everything, we don't expect the IKI test to be positive because the starch should have been digested by the amylase, converting it to simple sugars. You get that? So there should be no color change. Okay? It's a little bit confusing right here. Okay? What about here? There is amylase, there is starch, but you incubated it at 0 degrees Celsius. So what happened to the amylase? You're giving me positive again. What happened to the amylase? It's basically like you boiled the amylase. Okay, amylase will not work. So if you test the tube after 1 hour of incubating at 0 degrees Celsius, what will be your result? IKI test will be positive or negative? Positive. You get it? A little confusing, I know. But like if the amylase didn't work, so starch will stay. So what will be when you test it, there will be positive for starch. So
Negative,
that means
positive.
it is positive.
Negative,
The color
negative.
is blue-black. Okay? It's
Negative.
better for you to put the color here. If it's blue-black, it's positive for IKI. Okay?
Because I have a question. It's confusing me a little bit. But how it's
I
gonna
know.
be if about zero? That's been like it didn't have any chemical reaction because you need heat, no?
Where are you?
And the 6A.
6A, okay. The temperature is 0 degrees. Temperature. This is temperature.
Yeah, that's mean it's gonna be reaction. Like again, the amylase is gonna start digesting the starch, or not gonna happen because like it's low temperature.
Don't say no temperature. The temperature is 0 degrees. Well, you say when you say 0 degrees, it's like freezing. 0 degrees Celsius is the freezing point. Okay, it's cold.
Yeah, I know. That's mean not gonna have any chemical reaction here.
Correct, because it's like you are also killing the amylase. Because we said amylase will work at the optimum, meaning it will be working well at 37 degrees Celsius. Okay? So now you soak it for 1 hour at 0 degrees, you froze it. So it's basically, it will not be working well because it likes the 37 degrees Celsius temperature. Okay? You may not kill it, but like basically you are denaturing it also. Okay? Based on your, like as I was mentioning bio one, the temperature effect of temperature on enzyme activity. Okay, so it's like this one, you boiled it to high temperature, you killed it. You froze it, it's the same thing because enzyme will work at 37 degrees Celsius. Okay, is it clear? So I prefer you can put positive or negative, but give me the color change. Is it blue, black, or no color change? Okay, for this table, so you will not get confused. So let's do it again. IKI test for this one: amylase and water, negative, no color change. What about here? Starch and water.
Professor,
Yes.
do we just write no on that table, on that column?
That's what I was saying. You put positive or negative. Of course, when you say positive, it should be blue, black. You put blue, black, positive. Yes. Okay. Blue, black. Put the black positive. So here, is it blue, black? Amylase and water? No, right? What about here? Starch and water. IKI. Starch is there. This is blue, black. Okay? Blue, black. Next, maltose and water. Negative. No color change. What about this? Amylase and starch. You test it for IKI. Of course, there is starch there. It's just because amylase didn't work. So this is blue-black here, positive. What about here? Amylase and starch? No color change because amylase work at 37 degrees Celsius. So it should have dissolved the starch. So there is no more starch here. So IKI test is negative. Okay. Did you get the clarification? Negative, no change in color. What about here? Amylase and starch. You kill the amylase basically at 0 degrees Celsius. So you test for IKI. What happens here? Positive, because you did not dissolve the starch. So it is blue-black here, okay? So if you don't listen to me, I did it 2 times. You decipher it yourself. That's why I told you have to listen to me. Don't sleep. Okay, so here, well, the signs are just like positive or negative. So if it's blue-black, it's positive. If it's no color change, it's negative, okay? What about Benedict's? What is Benedict's test? Benedict's test, if IKI test is for starch, Benedict's test is for what? You don't remember? Bio 1? You remember. September. Benedict's test is a test for simple sugars. Okay? Simple sugars. If IKI is for starch, big sugars, big time sugar or starch or carbohydrates, Benedict's test is a test for simple sugar like sucrose, glucose, maltose. Okay? All right. So what do you expect here? Oh, before that, Benedict's test. What are the color for positive Benedict's test? It's a lot. It's not really a lot, but there could be different colors that signifies positive Benedict's test. So Benedict's test is a test for simple sugars. If these simple sugars are present, there will be a color change. All right. Now, if for positive for Benedict's test can either be green, yellow, orange, or you can cancel orange, green, orange, orange red, rubric red. Okay, those are the colors positive for Benedict's. So it depends on how concentrated the simple sugars are. Okay, if there is more sucrose, getting more and more, it's gonna be darker in color. So the lightest one, but it's positive. If it's positive for Benedict, it's green. Green to orange to red orange to rubric red. Okay, so here, let's go. Amylase and water. Soak at 37. What will be your Benedict's test? Will there be color change? Well, basically this table is color, you say color change. What is the color change? Is it green? Or you say no color change, and then here is just positive or negative, okay? Just giving you big table, but basically they're the same. Okay, so amylase and water, Benedict's test should be no change because there's nothing there, okay? It's just the enzyme and water. No change, so the result here is negative. Next, starch and water, test it with Benedict's. What will be your, let's say we are expecting a very good environment, as I tell you, it's a ideal environment, which never happens. Anyway, so starch and water, theoretically, okay, starch and water, so with 37 degrees Celsius, or you test it for Benedict's, what should be your result? No color change, okay? No color change, negative right here. Okay, maltose and water. Maltose tested for negative. As we said, maltose is a simple sugar. Green, okay, you say green. You can say green, positive, moderate chains, okay. Maltose, okay, so it's positive for Benedicts. You can give their green color, okay, and the result is positive. What about here? You boil the amylase at 37, you soak it at 37 at 1 hour. So we test it for Benedicts. What will happen? The amylase didn't work. There is no change. Yes, you will not break down these starch. Okay, easy, right? Now it's getting easier because you understand what we are doing. So it's negative here. There's no change. What about amylase and starch? Soak at 37. Benedict's test. Will there be simple sugar there? Yes, because amylase is working well. Okay, okay. You can say moderate. Okay, you can put green, orange, or put color in your chart. Okay, and positive. Okay. What about here? Amylase, soak at 0. What will be your Benedict's test? No chains, no color chains, okay? Negative, all right? So that's what you will do for the carbohydrates. So that will be your um, uh, salivary amylase digestion. Okay, so you just like for you to be able to give a good report, you have to read your like what I told you is basically what's in there. But for you to uh, make a good report, you will have to read your because that's the assay already. You see, if you look at your manual, that's what we call amylase assay. What we did in testing those IKI and Benedict's. Okay, these tables are the summary of what should have been done. Okay, so your manual will give you a more detailed description. Okay, so now let's go to the next activity, protein. You understand, right? Lou.
Professor,
Yes,
I have a question regarding that report.
sure.
What do you expect? Like, do you want us to write in a essay format or?
Yeah, you have to write like you just follow the especially for the discussion of what you do and for the methods. Okay, for the methods. Okay, of course you should have a title page, your first page. Okay, your title page should have your name, of course, and then the title. What is this? The title again, as I said, this is chemical and physical processes of digestion. Okay, chemical and physical processes of digestion. Okay. So then introduction. So introduction, what did I tell you? Like this is all about the food that we take in. What happens to the food? You can also put that burger, carbohydrate is there, the protein is there in the meat, the fat is also in the meat. Okay, of course, like from the meat we